delighted to be with you today. We have a great guest today. Isaac Galatzer-Levy is the Chief Scientific Officer at AI Cure. In addition to his role at AI Cure, Isaac is an adjunct professor in clinical psychology and is also a professor at NYU in bioinformatics. Before joining AI Cure, Isaac worked at Mindstrong, where he was recruited, I just learned, by Dr. Tom Insel, who used to be the director of the National Institutes of Mental Health. Isaac, welcome to Chet Talks.
Thank you very much for inviting me. I really appreciate this opportunity to talk with you.
You were a budding and promising academic, even had an NIH grant. What led you to ditch academia and go into pursuing digital measures of health? So I was at NYU full-time. I had a pretty promising academic career ahead of me. I was funded through NIMH, also receiving funding through the Department of Defense, and doing a lot of interesting international work. And I was running my grant, which was focused on blood biomarkers to predict post-traumatic stress response. So we were recruiting people in Bellevue's emergency room and following them longitudinally and trying to predict the course of who will develop PTSD and who won't. So that's a very important problem to solve, because only about 10% of people after a major stressor will develop post-traumatic stress or depression. And the ability to spot those at high risk allows you to best route the resources you have available. So that was a big focus of my career. And my early career award was going really well, and it seemed very promising that I was going to be able to advance to the next stage. So I was in the process of submitting an R01 to really advance this work, and I was trying to understand how to actually track the clinical course of a population, because we had spent so much time focusing on how to model the biological predictors, genetics, epigenetics, imaging targets, but our clinical definitions really hadn't changed for about 50 years. So at the end of the day, as somebody interested in mathematics and computational modeling, I knew that, anywhere in your equation, if you have error, you're not really able to find a relationship. So in the context of PTSD, for example, the way we define the disorder is through a hodgepodge of different symptoms that can be combined a lot of different ways, which produces really untenable heterogeneity. So for instance, in PTSD DSM-5 definition, there's over 600,000 different presentations of PTSD if you combine the symptoms different ways. So if you're trying to look at predictors, or you're trying to look at treatment response, you're really going to be very limited. So I was very attracted to the RDoC ideas around creating more specific clinical definitions that map onto our understanding of underlying neurobiology. That's how we better understand treatment and prediction. And I was literally about a week from submitting my R01 when my program officer recommended I reach out to Tom Insel at Mindstrong and ask if I could use the app they were developing in my upcoming study. And she connected me with him, and I talked to him on the phone, and he said, he talked to me about my research, and he said, to be honest, what you want to do with your career, I think you would have more impact if you came out here and worked with us. And why don't you join our team? We just have launched this company. So I went out there. I think I was employee number 6 at Mindstrong. I was originally recruited primarily to be in the data science team, so to be building clinical models and digital biomarker measurements of cognition. I ended up working in a lot of different areas across Mindstrong. But yeah, it was a very, very exciting time to go out there and learn, really a suite of tools. So for those who don't know, Mindstrong develops digital measures for a wide range of psychiatric conditions, especially around depression and others. What did you do at Mindstrong?
So my first job there was really to work on our digital biomarker models. So what we were doing was we were capturing keystroke activity, so the rate of texting, amount of errors, things like that, really the human-computer interaction with the cell phone as the method to track cognitive and emotional functioning. So when people are under stress, their cognition is impacted, their mood is impacted, and the hypothesis at that time was that it would be reflected in how they interact with technology. Human beings now are constantly interacting with technology. Really what our cognitive and psychological tests are is how do we interact with, say, a human being or paper and pencil.
And so what we were building were these models to basically track cognition in a passive way using cell phone readouts and identify when patients, for example, are at risk for relapse or likely to be hospitalized. So originally I was building those models, and then I moved on to helping to design the product around that, going out to clinics to understand, you know, how patients would actually use it, how clinicians would use it. So really end to end, how do you take a model that's promising and turn it into a technology that can really impact the patient?
What measures of cognition were most interesting or most informative?
So we saw a lot of effects, a lot of rapid changes in just working memory, executive functioning. You know, we saw that with patients as they started to become stressed, as they started to relapse, for example. We would see changes in cognition, and then that would cue a clinician who would have access to a dashboard to then reach out and interact with that patient. The next thing that we really did, yes, sir, go ahead.
And what were those changes? What were they on a smartphone? Was it changes in text? Was it changes in voice? What were the changes that you were observing?
So it was all based on, you know, our primary data source was all just text message. So it was just literally how fast were you text. So we created a very large number of features out of how you used your cell phone. So how often you hit delete, how many errors you made, how many, you know, how your rate of texting, which changes all the time. Those turned out to be very accurate proxies for many different domains of cognition. You know, the other major task we had was actually to build some, you know, chatbot type functions to actually interact with patients over a smartphone, over text. So, you know, as patients were distressed, they would reach out to us, and we would want to automate as much of the early interaction to assess what was the significant problems they're having, so we could best route them to the clinician that they needed. So I was involved in some of the early development of those, you know, those clinical decision-making, automated AI-driven clinical decision-making chatbots, which now are very much the core of the company.
Why'd you leave?
You know, I left for a couple of reasons. You know, so when I went out there, it was an extremely exciting and intense experience. You know, I went from being a clinical researcher who was good at building computational models, but in effect, I would have a static data set and I'd build, you know, some result and I would publish that paper and I'm done with it. And then I went out there and really my job was to build a scalable software where you're pulling in data at a continuous rate, passing it through models, having decision-making on top of that, routing it to clinicians. So I was learning an enormous amount about how you actually build a scalable technology to impact patients' lives. At that time, computer vision models were really taking off. So computer vision are models that allow you to analyze visual data. Going back about 6 or 7 years, their accuracy was about 70% to predict if something, for example, is a chair or not. When I was out in Palo Alto, everybody was playing with these methods because a number of new breakthroughs in machine learning had happened that had facilitated very accurate coding. And so I had been very interested in what are the visual and auditory signs and symptoms that we already know about. Can we code them? So for example, facial tremor. We know what facial tremor looks like. Can we build a model of that? If we can, then we can have a very accurate readout of a clinical interaction. You can model other types of neurological and psychiatric symptoms. Like a good example is eye blinks. You know, eye blink rate and intensity is impaired in a number of different disorders. But if you ask a clinician to count the number of eye blinks that during a clinical interview, that's extremely labor intensive and probably impossible. So I really liked the idea of, instead of replacing you know, the sort of clinical understanding that we already had with new signals, you know, maybe there's a signal in how you text for cognition or for emotion. What are the characteristics that we already know about, that clinicians are already trained to observe? Can we build models to support their measurement? So, you know, as a clinician, you interview a patient, you ask them how they're doing, and you see on their face, you hear in their voice how they're doing. You know, I can see a depressed patient, I can see somebody with psychosis. It's very easy to identify. Can we build models to support that?
So rather than tell us, you want to show us a little bit about the work that you're doing at AI Cure?
Sure, I'd be happy to. So I'll show you just a little bit. You know, this isn't a fully formulated slideshow, but I'll give you a couple of examples. So what we do is we deploy simple little tasks over a cell phone so patients can do them. We work with, we do this in clinical trials quite a bit. So we work with sponsors to understand when and how they want to assess. And we can ask open-ended questions like, how have you been doing in the past few hours? Just to get an open-ended response from them, both in their face and their voice. We show them different types of positive and negative images. We can ask them, for example, to smile and hold it for 5 seconds to measure tremor or psychomotor retardation. And then we use that to capture a number of different parameters. So here, I'll give you an example. So this is just a model of facial tremor. So this is a patient with tremor. This is actually taken from YouTube. And what you can see is the tremor. This is in their right and left lip and their jaw. We see quite a lot of tremor. And then their nose and upper lip, we don't see much tremor. Here is here's an example of voice tremor. Many of these methods are very well characterized. We do what all scientists do. We look at the existing scientific literature, what's been validated, and then try to spin it up and validate it for collection over a cell phone platform. Here's actually measuring asymmetric masking. So what you're going to look at is the difference between the left and right side of Muhammad Ali's face, his mouth, eyebrow, and eye. And this is the asymmetry between the two. So you can measure the different sides of the face and different landmarks to capture those types of characteristics.
Oh, you gotta go back there, man. That was the greatest. That's the greatest there. So you gotta walk us through that a little bit. So it looked like Muhammad Ali's right face was far more expressive than his.
Well, here, I'll show you this one where I'm going to compare him to 30 years ago. So here's a healthy person on the left. They have very little facial tremor. On the right, we see a lot of tremor. This is actually Deshana who built these models. Vocal tremor, here's somebody without tremor. And we can see a significant difference. And then here's that example. So here's Muhammad Ali actually talking about the same fight 30 years earlier. And this is the asymmetry in his face. And then here he is 30 years later talking about the rumble in the jungle. And we can see a marked increase in the asymmetry between the two sides of his face.
Whoa, you gotta walk us through that. So I on the video like really fast. Maybe if we can slow that down. And then what am I supposed to be looking at the graph?
So I don't know if I can slow it down, but what you're literally mapping here is how much does his mouth on the left side and the right side move together? How much does his eyebrow on the left and right side move together? How much do his eyes move together? This is a known symptom in Parkinson's, which is asymmetric masking, where one side becomes more flat than the other. Over time, you get overall masking, which you can also measure as just literally the amount the face is moving. So these types of measurements are very appealing to us because these really get to transdiagnostic neurological symptoms that are relevant in a number of different disorders. So our ability to measure the amount of expressivity or activity on someone's face, we see effects in major depressive disorder. So as patients get healthier, they actually increase in their amount of expressivity. As a clinician, that's probably not a surprise. In Parkinson's, you get this masking. You also get tremor. So I'll give you an example. Actually,
so hold on, just stay on here, Isaac. So is one showing would be perfect symmetry, and then scores above one would be increases in asymmetry?
Yeah. So as the score increases, we don't have a cutoff for what is healthy and not healthy asymmetry. We don't have those definitions yet, which I think is a very important reason why we have to be collecting this kind of data. Because we have definitions for cutoffs for sickness and health based on clinical rating scales. So I can say, on the HAM-D, if you're above this score, you're depressed. That we've defined those cutoffs. We really don't have cutoffs yet for things like actigraphy. What is a cutoff on actigraphy that tells me the patient is depressed? Or what is a level of asymmetry based on a model that tells me that this is unhealthy? The way that we use these kind of markers, particularly in clinical trials, is as a within subjects variable. So you can ask the question, does, for example, the amount of facial expressivity change in response to treatment, or does the amount of vocal tremor change in response to treatment? So I could give you an example actually here. These are patients with essential tremor who are on a medication and being evaluated longitudinally on the TETRAS. And what you're looking at here in green, This is actually our model of their vocal tremor, and we see a significant decrease. And what you see below it is actually the overall TETRAS scores for clinician rating and decentralized video rating. And what we see is that we get a significant effect for vocal tremor decrease on this metric, consistent with the overall TETRAS. You know, another
so
kind
just,
of—
we gotta walk through people. Isaac still has academic routes there, and I'm a neurologist, so I understand a little bit of this. But so essential tremor is a disorder that's characterized by an action tremor in people's hand, head, and voice. And what you're showing in green is that their voice tremor improved over the course of this study.
That's right.
In orange was their score on TETRAS, a rating scale, which really showed minimal change.
That's right.
That video assessments of voice tremor, for example, are more sensitive than clinician-derived rating scales.
Yeah, so I'll talk this through slower to your point, Ray. So what you're like, yeah, exactly. So green is our model. These are taking videos, that decentralized rater videos of patients in an active treatment for essential tremor. And what you're looking at is our model is in green, so that's the decrease we see based on our model. In blue is actually the in-person raters. So they're more sensitive than what you're seeing in orange, which is the videos are sent out to a reviewer who then scores them independently. So what we're seeing is that the in-person rater is more sensitive than the remote rater, and our models are actually more sensitive than both because we can directly quantify the amount of tremor. And I think this is an important point about measurement. You know, we lose a lot of sensitivity because our measures are not actually real numbers, they're ranking scores. So if I say somebody has a tremor on a scale of 1 to 4, that's a ranking. I lose a lot of sensitivity that way. Now, if I'm actually quantifying the amount of tremor on someone's face or in their voice, I can create a real number score, which is by definition more sensitive than a ranking. And so we're going to get more sensitivity just by objectively modeling the data rather than depending on a clinician's interpretation and putting it on a scale that doesn't have a real zero.
So a couple things, you're going from a categorical rating scale to a continuous measure with your video rating. In addition, your video rating is independent of a clinician. This is the computer making the rating completely independent. Right. Is that correct?
That's right. That's right. So we're modeling the tremor by actually looking at changes in facial position from frame to frame. So when we're measuring with video, we capture at a rate of over a cell phone, we can capture between 30 and 60 frames per second, depending on the technology. So at that rate, you're moving almost as quickly as the actual physiology itself, which means you really have no data loss. You really are able to actually measure correctly every moment of tremor on a person's face, compared to a clinician who's sitting with a patient trying to observe tremor while taking in a lot of other information and then putting it on a scale that isn't a real number scale. So there's some inherent limitations in that type of measurement that can be overcome with these kind of models.
Great, and we're getting some questions in our Q&A, so please put the questions in. If you want to go geeky, Isaac is happy to go geeky, and if you're more like me and you want to say, what does this all mean? Feel free to put it in English too. Continue on, Isaac.
Yeah, absolutely. So here, let me just show one more slide, which is in MDD. So this is patients recruited for major depressive disorder, exactly, and they were recruited and then did a course of standard SSRIs. And what we did is we followed them longitudinally. They were assessed on the MADRS, which is a standard scale for depression, which you see in red. The patients are decreasing in their MADRS scores. What you're looking at in black is 3 measures of facial expressivity. This is just literally how much is their face moving. So once again, in major depressive disorder, we get a symptom known as psychomotor retardation, really these underlying motor abnormalities that come with depression, where people are very much slowed down, their voice slows down, they talk less, their face moves less. And so what we see here is significant decrease in the MADRS, which maps on with significant increase in facial expressivity when we show people positive, negative, and neutral images. We see the same thing in terms of
Hold
pattern.
on. So stay there. So the neutral image, like positive image is a flower, and neutral image is
Yeah, a positive image could be, a parent with their kid smiling, something that evokes a positive response, has been standardized to evoke a positive response. A neutral image can be something like, a picture of the coral reef that doesn't really have much register. A negative image could be something like a violent or disturbing scene that tends to provoke negative responses. And then we can look at just literally how much do people respond. So human beings, we respond with emotion constantly. We show different facial expressions all the time. So I can tell how interesting I am right now by reading Ray's face, right? This is just a human activity. The first person to actually write about human emotion as a biological process that's meant for communication was Charles Darwin. It was his second book after On the Origin of Species was on human emotion as a evolutionarily evolved characteristic that is meant for communication. So it's not surprising it's easy to register and measure directly. Now, what I like about this type of measurement is that it really starts to let us move beyond these categories like depression or Parkinson's or, and really start talking about underlying, pathophysiology of the disorder. So we see that psychomotor retardation in many different disorders. We see tremor in many different disorders. If we can monitor that correctly, then we can start thinking about our treatments also transdiagnostically.
So if I'm reading this again, is that person with depression, you started them on an antidepressant, the rating scale in red got better over 5 weeks. And correlating with the improvements on the rating scale were increases in expressivity in response to a variety of images over the same 5 weeks. One requires a trained rater to do this assessment. The other one requires nothing but a smartphone.
Exactly. So we capture it takes about 2 minutes. It can be done remotely over a smartphone. Exactly. So another example of that here, this is just how much are people talking? So we ask them this open-ended question, what have you been doing in the past 2 hours? Just to get them to talk. And we're looking at literally how much space is there between words. So depressed patients also have that, their speech slows down quite a bit, also due to that same psychomotor retardation. So they literally talk slower. And so we can count the white space between words. And what you're looking at here is that same MADRS score going down, and then what you see going up is literally the rate of speech. So the patients are increasing in how fast they talk in response to the SSRI.
So I'm going to slow you down here. The y-axis is, what's the y-axis here?
So the y-axis here, we've created a normalized score so that we could put these on the same scale. So, rate of speech, we could do a simple count, but the MADRS is on I think a 1 to 30 scale or something like that. So they are sort of, to make the graph look correct, we normalize the two scores.
But what are you counting for speech? Are you counting number of words per second? Are you counting duration of pause between words?
Yeah. So it's a ratio of the pause to actual speech. So what's decreasing is the pause between words, and so the amount of speech in some segment is increasing.
And can you count the number of words somebody speaks over a recording?
Yes. So it's actually pretty trivial at this point. Natural language processing and recording voice is very, very accurate now because any of you guys who have Alexa or any of these connected devices, they're used to train models. So now we can very, very accurately count words. We can very accurately transcribe language for analysis. Many of these methods, we haven't invented at AI Cure. These are methods that are out there and are accessible. So yeah, you can literally count words. You can code them for emotional valence. So you can, once again, in depression, you could say, or Parkinson's, anything with mood symptoms, you can actually measure what is the valence of someone's speech and how does that change in response to treatment. So many of these methods are very accessible and even off the shelf at this point.
Well, perfect. I got a research project question for you after Chet talks,
Okay.
but continue on.
So So yeah, so that is the general approach, is to say, what clinicians do is that they interact with patients. A patient comes to you, you ask them how you're doing, you collect an enormous amount of information because you have a model, you have a psychological model that you developed in your training, and you say, oh yeah, that person, so I worked in a state mental hospital. You could very easily identify a psychotic patient who's acutely psychotic. Their language is confused, their speech is erratic, they look agitated, so they're moving a lot. These are characteristics that we can directly measure now. Similarly, on the other end of the spectrum, we can measure slowing. So one area we do a lot of work is in negative symptoms of schizophrenia. Once again, you get very flattened face in schizophrenia. To my previous point, the mechanism is much the same, right, across these different disorders. And so by moving to outcome measurements that are transdiagnostic, we can start to look at underlying biological mechanisms that really map across disorders. And so treatments can be relevant across many different disorders. So it's not a surprise, many of our drugs are used off-label for other indications. Antipsychotics are prescribed in people with depression quite commonly, and it's because these categories are manufactured, but the direct symptomatology that clinicians observe is real and cuts across disorders.
We're getting some questions about the rate of change of these markers. There seems to be a fair amount of standard error, so kind of hard to tell which one's more sensitive or less sensitive. But in general, do you find that your digital measures are more sensitive to therapeutic effects, or are the paper and pencil rating scales more sensitive to these pharmacological effects?
Yeah, so it sort of gets into a tricky problem of what does, what are the rating scales measuring, and then what is sensitively changing, for example. So in major depressive disorder, for example, if you look at a standard measure of depression, let's say the HAM-D, you have a set of symptoms that mechanistically may not even relate to each other. So for instance, you have mood symptoms, you have sleep symptoms that can go either direction, hypersomnia, insomnia, you have gastrointestinal symptoms, you have guilt symptoms. And so what changes in response to regulation of serotonin? Which one of those markers really makes sense changing in response to serotonin? Some may not. So if you have a measure that's very heterogeneous, it's very hard to actually identify change, because you may actually be very accurately targeting certain symptoms and not others. And so overall, your effect washes out. So PTSD, where I started my research, is a very good example of that. Because the diagnosis is so heterogeneous, because we've included so many different symptoms, because in effect, a lot of bad things can happen to you psychologically after a traumatic event, it's very, very hard to track change. Because you may actually have significant change in areas like arousal that have some specific underlying neurobiological substrates, and not changes in mood symptoms. And actually, I understand mechanistically how to affect arousal and how to affect mood, and they're not necessarily, they're overlapping to some degree, but then somewhat not. So if I can better understand what it is I'm trying to target, I could actually select people better for treatment, and I could actually track the effect of a treatment. So the question of sensitivity is sort of an interesting one, because what is sensitivity to what? The definitions of psychiatric disorders are, really they predate the biology that we're trying to manipulate, or our knowledge of the biology we're trying to manipulate.
We're getting a couple questions around privacy, not surprising. People are knowingly giving you these videos, I assume. I assume these are being reviewed by IRBs. Muhammad Ali's video, I assume, was in the public domain. Did you get an IRB review?
So all the demos we showed here, we used YouTube videos. So YouTube may sue us, but we did it so that we did to step around PHI issues. Now, importantly, PHI is a really major issue. You're capturing video, which is very risky. So I can claim no credit for this. This is really, one of the major reasons that I came to AI Cure. They were interested in measuring, digital measurements of clinical functioning to look at dose-response relationships. So AICure was already measuring adherence using video. So they use direct observed therapy where patients, the app opens up when it's time reminds people when it's time to take their medication, and they take the pill via the smartphone, and we recognize the pill and we recognize the ingestion using AI. That itself is a digital marker of ingestion. It's a proxy measure that tells us this person took the medication. Because that technology has already been built by AICure, and we're working with the majority of large pharmaceutical companies to track adherence internationally, we had already worked out, and not we, this was before I came to the company, had already worked out all the legal regulatory issues around capturing that kind of data, which really at the end of the day is a major impediment to this sort of thing moving forward. As I said, many of the models we use are accessible and off the shelf, but the ability to capture this kind of data, transport it, analyze it in a study happening in the Ukraine, for example, really requires some very complicated infrastructure that AICure had really figured out before I had arrived.
And people are giving consent to have their videos.
Oh, yeah, absolutely. Absolutely. So they're fully consented, they're read in. Oftentimes with these digital biomarker studies, we're invited to patient advisory boards ahead of the study to get patients' feedback about their comfort. Now, one thing that has been very interesting to me, working in both the area of passive data collection at MindStrong and active data like this at AICure, is that patients tend to be actually much more comfortable with these data sources because in effect, they are aware they're providing data every time that they do. So just like people are very comfortable telling their clinicians all kinds of things because you establish a rapport with your patient and they come to you and they're willing to divulge information, we very much think that collecting data remotely has to create a similar type of rapport. You have to create trust in your patient. You have to have them know what you're doing and why you're doing it. Now you get the benefit of capturing lots and lots of data and not necessarily, so it's harder to game it. But aside from that, we think that, that kind of active interaction, allows patients to have more buy-in, and we hear that actually in our user testing.
The Digital Medicine Society, our friends there at DIME, have created a library of clinical trials that are using digital endpoints as outcome measures. Even today, few clinical trials are using digital measures as outcome measures. Is that going to change in the COVID world?
I think probably everybody has noticed, really rapid change with the onset of COVID in terms of the use of technology in healthcare. These ideas that were really nice to haves a few months ago are now becoming essential. So my typical interaction, before COVID-19 was, the tech innovation guy that the large pharmaceutical company hired to figure out where the company's going in the future, who, the clinical trialists didn't want to really listen to because they knew how they wanted to do it and they didn't want to change it up. Those are the people who wanted to hear about these measures and wanted to experiment with them, but had a lot of trouble moving them into trials because, FDA doesn't recognize them yet. Validation is a long process. And they weren't very motivated to do things differently. What I'm seeing across these companies is those digital innovation guys are getting promoted. They have a more central role, and everyone's trying to figure out, how do I make my trial digital in a hurry? And FDA is doing a really good job of saying, we need to open things up and we really need to be progressive right now. And so all of those agents are converging at the same time, where a lot of the technology already exists. The same thing with telehealth. The technology existed, we were just not subjectively comfortable with it. So at NYU, for instance, we've had, virtual urgent care for like 3 or 4 years. And I myself didn't like using it. I felt it was realer to go and talk to a clinician in person. And all of a sudden, my attitude has totally changed. My daughter had a virtual, health meeting yesterday. And, once you're doing remote interactions with patients, the next obvious thing is, how do you measure patient functioning? How do you measure it accurately?
We're going to go to questions in just a second. Can you give an example of a study that's now incorporating one of your digital measures as an outcome measure?
Yeah, so we're actually in a number of different studies. We're in a study of, we're in a phase 3 study of Parkinson's disease. We're recruiting both early and late Parkinson's patients to look at some of these more sensitive measures like facial and vocal tremor. We are in a study of major depressive disorder also that is actually a completely virtual trial. So they're using our technology and some other technologies. So really, we're ahead of the game in terms of how do I make a trial virtual. We're working in the area of negative symptoms of schizophrenia as well. But really, because of our focus on really transdiagnostic markers, we're able to go a lot of different places. So we're starting to develop studies in areas like autism, areas outside of traditional neurological or psychiatric disorders, but that have CNS side effects, like say stroke or even oncology. So really, if you're thinking about, what is the output of the central nervous system in terms of behavior and physiology, there's an enormous range of areas where either the primary effects of the treatment or even side effects are really relevant to measure digitally.
And it sounds like for depression and schizophrenia, are you using facial videos? Is that the outcome that you're using?
And voice. So we're analyzing so we really do 3 different things. We analyze the video for facial expressions, different emotions, their intensity, movement parameters. So how much do people literally move around? So for instance, people with negative symptoms of schizophrenia stand very still. They don't move very much. We also measure voice measures like pitch, tone, rate of speech. And we also analyze the natural language. So what is the valence of their speech? So patients with schizophrenia use a lot more negative language. Their negative language is invariant to context. So if I show a negative image and ask you to describe it, you talk about it in a negative way. If I show you a positive image and describe it, you talk about it in a negative way. That's, if anybody's ever interacted or had depression, that's not unfamiliar. So really not variant to context. So can we create that context on the phone and measure it?
Rachel Francine asks, how are you measuring vocal tremors in your current study?
Right. So there's a number of different, the short answer is our approach to measuring vocal tremor is probably not different from most people who are trying to model vocal tremor. So there's a number of different features that are relevant to capture a vocal tremor, and then we combine them into a multivariate model that increases accuracy. So we can measure pitch, tone, any number of other vocal parameters, and then train a model to match it to level of tremor. We can actually experimentally evoke different types of tremor. So for instance, you can put a device that tremors someone's throat to actually create tremor at different frequencies, so they create a real number. We did the same thing with our hand tremor models. So what we initially did, because we thought that the clinical scores would be challenging to train against, we actually had healthy people and we passed different frequencies of electricity through their hand to increase the tremor. And so we knew the real voltage, and so we could train against that as our sort of base model. And then we actually improved those models against real clinical scores.
What other populations do you work with besides depression, PD, and psychosis?
Yeah, so we're very quickly moving into a lot of different areas, both in psychiatric and neurological conditions and areas outside of psychiatry and neurology. We're building models in a lot of different areas very quickly. A good example is just in the last few weeks, for obvious reasons, we spun up models of cough frequency and intensity. Once again, it's the same vocal features. We train up a model and we validate it. And already people are interested for a number of different indications. We're working on physiological models. I know you have some colleagues at University of Rochester doing similar types of work where you can model actually using video. You can measure things like a heart rate or respiration by looking at breathing patterns in someone's chest. So we're working in a number of different areas and a number of different indications. And we've only been at it for about, I've been at AICure about 1.5 years. So really, it tends to follow a pretty predictable path where we have a set of models and then we validate it against some clinical data in a certain indication, and that leads to a number of different sponsors being interested in using those as endpoints.
So shout out to Dr. J.P. Kuderik, who's shown that video can identify atrial fibrillation by looking at changes in pulsations in the face, and shout out to our colleague Asan who's developing a video analytics platform for assessing Parkinson's disease. On that note, Marvelin DeCambre, apologize for any mispronunciation, says, do you know where Muhammad Ali was in that first picture? And where was that in relationship to his history of concussions?
So the first one, I actually knew this. I'm trying to remember. So both of the interviews were him talking about the Rumble in the Jungle. And I think that one, the video was actually before the fight. So there was it was an interview about the fight that was going to happen. And then, I can't tell you exactly where he was in his disease progression, but I can say it was earlier.
And so the Rumble in the Jungle was Muhammad Ali fighting George Foreman, a young George Foreman, a very dangerous young George Foreman. And I think it was done in Zaire, if I'm recalling.
That's right. And
correctly.
George Foreman was, the problem, if anybody remembers, is George Foreman was slow but an incredibly hard puncher. And so that's where he invented the rope-a-dope, right? Where he leans back and let George Foreman punch him so he trained up his stomach to be able to take a lot of hits and wore him out.
And so if you want to learn about concussions and Parkinson's disease, a shameless plug for our book, Ending Parkinson's Disease. It talks about Muhammad Ali. It talks about concussions. It talks about the risk of Parkinson's in football players, among other topics. endingpd.org, and we have a whole series of webcasts on it. Follow-up question, same person. Is there an argument for in-person rather than virtual visits for some of your technologies?
Yeah, absolutely. One of the things that people are really interested, with these technologies is using them for screening and really to confirm screening. So that example that I showed you of, how does the in-person versus the remote rater, how well do they compare to each other? This is kind of a thing we've known for a while with ECOAs, that remote raters, they lose a lot of the nuance and their scores tend to be a little less subtle. So, one of the things that we've actually been doing quite a bit is just, rescoring in-person video ratings. So, can we confirm and use that for, say, screening? So the clinician said, this person had this much tremor. What does your model say? We're trying to make a decision about, whether to enroll this person or not. There's many instances where people don't want to use the smartphone platform. So a good example is, we're approached occasionally about studies in autism with children, where you can look at characteristics like, once again, facial characteristics of emotion, attentional patterns, people's eyes, where are they going? But those tend to be with a clinician, a type of interaction, and tend not to work well over a smartphone. So we've really built the capabilities to take in videos and analyze them the same way. I really think about the models we've built, the back end, the engine, really to be the important part, because that's, the ability to take video and audio, pass it through a set of models and output clinical insights is really the value I see.
So Isaac, we're going to go to the speed round now. I know you're not a speedy guy, but we're going speed round because the questions are coming in.
All right, I'll go fast.
Speech for sparseness in speech. How do you distinguish the individual's voice from other speakers?
Yeah, so this actually gets to most of the problems with collecting remote data, is not so much the models. Because like I said, a lot of these models are out there in the world. It's really, how do you deal with those types of problems when you really are not observing directly? So we have to do things like use algorithms that can differentiate one person's voice from another, or separate a voice from background noise. When you're dealing with patients, especially psychiatric patients, they don't necessarily follow all the rules. So you may have patients who are talking while the TV is going in the background. How do I know, am I measuring your mood or am I measuring the mood of the character on the show? These are big differences. So we have to do a lot of data cleaning. Same thing with the facial characteristics, actually. If you're not in enough light, I can't measure anything. So I have to be able to detect, I need to be able to prompt a patient to do the right things for the assessment, because I really don't have a clinician or a human in the mix.
John Gurdrow asked that exact same question. How dependent is the face expression data on background lighting, distance from camera to face, camera quality, and other distractions?
Yeah, so there is.
How do you standardize?
Yeah, so there is some real pluses to cell phone-based assessment. The native way we interact with a video on our phone is literally about 6 inches from our face on. So that allows us to actually do a lot of pretty impressive things. So for example, we can track pupils well enough to know if somebody is looking at their screen or not. So we can measure, for example, attentional patterns. You know, in patients with autism, are they, if I show a social scene versus a non-social scene, how much do they look at that versus look away? I can do that kind of thing because the native interaction is actually my friend there. But we also have a lot of built-in sensors and a lot of built-in methods to actually improve those measurements already native to your cell phone. So, right now when you're, on your cell phone, you're not seeing a lot of movement artifacts, and that's actually because of algorithms that are native to iPhone or Android. But, additionally, we have algorithms that we've built at AI Cure, for example, that make you have your face in a frame. And if you move it out, it will tell you move your face back into frame. And those were really developed for the adherence platform because we had those kind of fundamental problems already.
You're not very good at the 30-second rule, but we'll get you better.
Okay, I promise.
Matt Eagles, are the measurements taken once a day during the example for Parkinson's? Some of these diseases change in seconds, or do you have to follow a video record them all day to get a true reflection?
So we don't video record all day. What we're doing right now in the Parkinson's trial that we're in is we actually are allowing patients to turn it on when they're in an off period. So so we measure twice a day because there's variation within the day, of course. But we also allow the patient to just, when they're having their worst incident, to actually turn it on and do the assessment. We see that as a big advantage over, in clinic where you really are just stuck with whenever they come in.
Thomas Sablinski says, great work, AI Cure. Can you define digital biomarker, short and simple, in a way that can be understood by laymen like VC investors?
Yes. Oh, so digital biomarkers is a catchy phrase, but really what digital biomarkers are is a digital measurement of a biological phenomena. So I would say that
digital measure of a biological phenomenon.
That's what I would say.
Beautiful. Mr. Shaw, I think, how does this technology work if the video is choppy or the internet is weak in developing countries?
Yeah, so what we do at AI Cure, this is a short answer that is actually a solution anyone should use, is the app actually captures the video even if you don't have Wi-Fi and then uploads it when you have Wi-Fi. So you'll have the same problem with Zoom, it'll be too choppy, any kind of thing that's on real time. So what you need to do is build an app that actually captures the video and then uploads it.
Leah Holman asks, what's your policy on sharing the models you developed? Are they published where other researchers could potentially deploy them?
Yeah, so everything that we develop, we try to publish. Publication isn't overnight, but we try to do that. Our goal is actually to make all of our models open source and available. We think that the only way science advances is to share definitions and share code and share knowledge. So really with all of these models, that is our goal, to create methods where our pipeline in effect is open source and people can evaluate it and add to it, hopefully. And
what's your secret sauce, or what do you guys, if you're making the code open source, what's your value?
At the end of the day, we have a very strong scientific team that can build these models. We also have the capabilities to deploy them, almost anywhere in the world in a clinical trial in a HIPAA GDPR compliant way. That's enormously valuable. So that's what we see, and we are able to help people understand the data.
John Goudreau, are any drugs or therapies being approved using a digital biomarker as a primary endpoint?
No. And not that I know of, I'll say, I won't say no, but not that I know of. And this really gets to, how do we validate these kind of measures and how do we move them forward? It's really quite challenging. So as far as I know, in CNS and neurologic disorders, there are no primary endpoints that are digital biomarkers.
So an MS drug was approved on a timed walking task done in
cool.
The EMA, the European Medicines Agency, has indicated that a gait speed measured at the 95th percentile has been approved as a primary endpoint for Duchenne's muscular dystrophy. I'm not sure if that's been done. And I've got a paper in my living room about looking at moderate to vigorous activity as a primary endpoint in congestive heart failure. So I think there are really not many great examples right now, but that could change in the next 6 to 12 months. We're going. Are you, Dr. Dicker from Jefferson, our good friend, asked us, are you using these tools outside of neurology and psychiatry?
Yeah, so we're just now getting a lot of folks who are interested in other areas, and this really has to do with exposure. So we've been working quite a bit with, a large CRO, to look at different areas. And so increasingly in areas like stroke and oncology, where you get a lot of interesting side effects. One indication, for example, that people were interested in was literally palatability when people are taking medications in oncology. Many of them are quite disgusting, and people, people gag. But this is just a straightforward disgust response. Paul Ekman, demonstrated how to code disgust, 40 years ago. So
only on Chet Talks do you find out that you can get a digital measurement of how a pill tastes. Only on Chet.
Exactly. That's right.
You said digital biomarkers are digital measures of biological phenomenon. Do you have any data linking the effects of what you're seeing to neural correlates in Parkinson's or Alzheimer's?
So there's a very large literature looking at characteristics like facial expressivity or movement, and then, functioning in the motor cortex, other regions of the brain, or neurotransmitters. We have not done any direct work demonstrating those links on our cell phone data to the biology, but there's a very robust literature in these areas.
And that came from Matt Betts. Edward Schwartz asked, has this technology been adopted across the racial and ethnic spectrum of humans?
Well, I don't quite know how to answer that question. I mean, I can answer the question about the generalizability of these markers. So, Darwin is actually the first one to talk a bit about the generalizability across age and different ethnic groups. His argument was because they're biologically conserved, we see that. Now, it's been confirmed that certain emotions are very much cross-cultural. So, for instance, the big five: anger, fear, disgust, sadness, surprise, are highly replicated across different ethnic groups and races. Same thing with kind of pitch and tone characteristics. Now, when you get into natural language processing, there's some very interesting things in machine learning that actually cut across cultures, but some of it is very cultural dependent. And the way we use language is very culturally dependent.
So we've gotten everyone from Charles Darwin to Muhammad Ali discussed here. Wrapping up, you mentioned that I think your daughter used telemedicine. Telemedicine is having its finest hour in the setting of COVID-19 pandemic. Virtual and decentralized trials may soon follow, and that's what we focus a lot on at the Center for Health and Technology. Are digital biomarkers next?
My opinion, 100%. It is the obvious next step. I talked to my wife, who's a clinical psychologist. She has a patient who's a neuro-oncologist. And she said, I'm just asking people about their tremor over Zoom. I can't measure it. If we're going to have remote assessments, why do we go into a physician so they can put a blood pressure cuff on us? We need, clinicians, what they do is they measure people and then they prescribe based on that. And so now that we have an interaction, you need to create a way for measurement, and that's got to be digital.
Isaac, thank you very much for joining us on Chet Talk and helping us see the future of digital biomarkers. On our next Chet Talk, we turn to healthcare without walls. On Thursday, May 21st at noon Eastern, we'll be speaking with Susan Dentzer, a senior policy fellow at the Robert J. Margolis Center for Health Policy at Duke University. For those who don't know, Susan Dentzer used to be the editor-in-chief of Health Affairs, the top health policy journal in the country, and was also a correspondent for the PBS's NewsHour. If you want to learn a little bit about journalism and know what the future of health policy and healthcare delivery will look like, join us next Thursday. Until then, the Center for Health and Technology at the University of Rochester thanks you for joining. Have a great week.
Thank you very much, Ray.
Thanks, Isaac.